To introduce Dr. Cardé. Ring's an esteemed insect behaviorist, and he's going to talk to us today about Aedes aegypti.
Ring. Can you see me, or are you just seeing the slides at this point?
Just see the slides.
Just see the slides. Okay. Well, I perhaps should give a little bit of a background as to how I got here and why I got interested in this. For many years, I worked on insect pheromones and then later incorporated insect parasitoids, and interested in both cases in how parasitoids and male moths find their quarry. And the idea of when I came out to Riverside was that the folks thought that it would be really useful if I could apply some of this to mosquitoes, and I was kind of willing because I thought it would be something that would readily translate. But in fact, it's a difficult problem. For one thing, if you, let's see, it's supposed to go forward. Oh, there we go. For one thing, in the case of moths, we have really simple systems. We have a couple of compounds. A male finds females very quickly. The wind tunnel assays is pretty simple. In fact, the chemistry is sometimes pretty simple compared to what we face here. In the case of human skin odors, there are far too many compounds. If you look at Uli Bernier's papers, you come up with several hundred, and there are yet more from human sweat, incubated sweat, human breath, all kinds of sources that might conceivably have odors that mosquitoes would have. And then you have three lineages of mosquitoes, which have been separated in a long evolutionary time. And there's no reason to imagine that they have to use the same compound, so that as we compare Aedes aegypti with Anopheles gambiae or Culex quinquefasciatus, we might find some overlap, but we might find quite a bit of difference. And when I say we know so little, I could have said we know so much, in that there are literally hundreds of papers attempting to sort of look at the whole question of how these cues are used by mosquitoes. And it isn't just odor cues, it's visual cues, it's heat cues, possibly humidity, and how they are sort of cascaded into response remains a bit unclear. So, if I go the right direction here, many folks have sort of approached this by using various kinds of bioassays, at least as far as the chemistry goes, and this is from a USDA lab. And mosquitoes are put in one end, and they have an opportunity to go to the other end over some long period of time. And clearly you can produce publishable results out of this, but it doesn't necessarily translate into anything that works in a wind tunnel, or much less in the field. And folks therefore have gone to some better representation of trying to understand natural orientation. And I still have to get this correct here. By using various, these are not really wind tunnels, they're port assay systems. The one on the left here is from the USDA in Gainesville, and it has an even flow of air, and it has two entry ports. The mosquitoes are released, and they can make their way into the entry ports. In the Netherlands, where they've studied Anopheles gambiae, they've used a similar kind of system, except it's a much larger box, but there's no airflow in it. You just have two ports that dribble out odors, and you kind of look at whether or not they go on one side or the other. And if you get positive, in the sense that you get a few going in one side, you sort of off to the races, and you perhaps try and use these things in the field. But there's a lot of misleading information that can come from that. And one of my former grad students, Ton Decker, looked at that by doing something that I guess they'd failed to think of, and that is to look at the time course of what happens. And in Wageningen, they normally run a 15-minute assay. When they run a 15-minute assay, at the end of the assays, you can see from all these lines, you get everyone coming into the port. But if you look at it at 3 minutes, you realize that some treatments are far better than others. And so this is one of the problems with many of the assays that you see in the literature. The latest one I saw in the literature, I think, was 7 minutes was their assay time, was with Aedes aegypti, and they said, well, that gave them the best results. Question is, if you have an opportunistic mosquito like Aedes aegypti that under some circumstances will come right up and bite you, have you mimicked what you'd like to mimic? And it's even more complicated, because if you look at that chart, There's another way you could do that, and that is you could put an electric net in front of the port, and then you get another set of interactions. And in fact, you find that some things that don't cause, some odors that don't cause the mosquito to go into the port, they actually cause the mosquito to hang out at the entrance of the port. So if you have that net there, you electrocute them, you realize that there's something that's going on. So what we did is to create a wind tunnel system at Riverside that enabled us to look at this. And just very quickly, you have air flowing from right to left. You have a series of patterns on the floor, which provide optomotor feedback, basically, to enable the mosquito to know what its position is. A release cage, and you have a couple of video cameras so that you can map their behavior in 2 dimensions or 3 dimensions. And we wanted to look at this in several different ways. The first thing we wanted to think about was carbon dioxide. Recall that carbon dioxide is present in the atmosphere at 0.04%, although it is rising, and we exhale at 4%. That's only a 100-fold difference. And mosquito maxillary palps, which sense carbon dioxide concentration, are exquisitely capable of detecting its very small changes in concentration. And here you can see a 1 second change in concentration, very small change in concentration that goes up, the spike rate goes up, and if the concentration is lower, the spike rate goes down. So we know that this is important. We also know that naturally plumes have turbulent structures, and I won't be able to go through a lot of data that we published on this, but just to give you a brief idea of what we can do, we looked at 2 kinds of plumes. We looked at sort of a broad plume that was fairly even, that is homogeneous in structure, carbon dioxide, and we also looked at a plume that was turbulent. And by turbulent, I mean we actually used a small glass ring with holes in it, and the carbon dioxide is piped out, and it produces a very, very structured plume that sort of resembles a wispy smoke plume. And what we find with that is that if you have carbon dioxide and it's homogeneous or turbulent, you actually get pretty good movement upwind if it's a homogeneous plume. If it's a skin odor plume, we actually find something very different. And that is that it seems that this mosquitoes favor a much more homogeneous odor from the skin. So I won't go through all of the details there, except to point out that this is just an interesting facet of it, and it also suggests that, at least in part, we understand that carbon dioxide needs to be presented as a fluctuating hue rather than a homogeneous signal. There's one other thing that surprised us a lot, and that is that the contact with carbon dioxide actually changed the threshold for response to skin odor. Now, what you have is the possibility in the wind tunnel of giving them a short pulse of carbon dioxide and then asking the question, how do you respond to skin odor? And what you can see here is that if you have a high concentration of skin odor, it doesn't much matter. But if you give them a little bit of carbon dioxide in advance, as you see in the green, you actually can get very, very good response. But if you just have the skin odor alone, you don't. You can actually go through a kind of dilution. People here can see me pointing, but I guess I'd have to. There's no mouse for me to do that here in this system. There is a mouse.
So I don't know if it'll show up.
Well, at any rate, the clear idea here is that carbon dioxide, in a sense, changes the threshold to response to skin odor. Now, of course, we are imagining that almost all traps will use some version of carbon dioxide release, possibly with some skin odor. And so, in a sense, we are already trying to do that with most of our trapping systems. So this led us to think even more about the importance of this issue. And one of the postdocs in my group at the time, Ben Webster, devised a fairly simple system for looking at another kind of behavior. Up to this point, we've been looking at orientation and not really paying much attention to landing. But as we all realize, that's the ultimate situation that we would like to monitor. Does a mosquito actually go up to you and land on you, and obviously then perhaps continue on to bite. So what he did was to ask the question, is carbon dioxide modulating that behavior? He was willing to go out and buy lots of pantyhose, which he could then wear, and that would get his skin odor onto the pantyhose. And then he could put that in a cage. Now, you can see the picture of the cage. These are awfully scanty, so we're having a, whoops, sorry. It's dark in there because this is a nighttime mosquito. But you can see on the right what you get if you are putting a little bit of carbon dioxide into the cage. You get landing on the skin odor, which you do not get, surprisingly, without this change in carbon dioxide. Remember, carbon dioxide is already there. And in fact, one of the things we found out is that in one of these bioassay rooms, if you walk into it, you start increasing the carbon dioxide concentration. So the protocol for almost all of our work now is you take a deep breath, hold it, run into the bioassay room, set up your bioassay, run back out, and exhale. And you can see in the data here just how profound that difference was in landing percentage. So this is run over 6 minutes, and you can see the concentration of carbon dioxide that was added. So we have up to 4%, which would be natural exhale. And if you don't have that, you essentially have no landing. So again, this is another profound effect of carbon dioxide on threshold of response. And it's really remarkable to me that you just don't seem to get any landing on this source unless you change the concentration of carbon dioxide in the atmosphere. So this then sort of summarizes a little bit of it in the sense that fluctuating carbon dioxide signals affect orientation and landing, and they lower the threshold of response to human skin odor in a couple of species. With that in mind, and we'd all been thinking about this, we concocted some sort of a generalized idea of how cues interact, and this is a little bit of a summary. If you start on the left, you have activation, and we're not really sure if this is something that occurs in the field exactly, or the mosquito may already be flying about, but in the lab, often the mosquito is quiescent. We don't really know how far away this occurs, but we've now done some calculations, and those suggest that if you have a human emitting at 4%, the volume amount of carbon dioxide produced, it will become imperceptible at least within 30 meters downwind, and probably a lot less than that. We do those calculations with a lot of atmospheric turbulent dispersion models, and we should probably do some more field measurements, but it seems that's probably the maximum distance. At some point, if a mosquito then detects an above threshold concentration of carbon dioxide, it presumably moves upwind. We believe, of course, based on all the work that's been done with flying insects, that this is the optomotor response. It requires visual feedback. You're not sensing how the air is moving mechanically, nor are you sensing a concentration gradient. But you begin to interact with other cues as you get closer. Visual cues, obviously. Mosquito that's landing has to prepare to land. It does that with a very distinctive maneuver where it spreads its legs, and depending upon the dispersions being horizontal or vertical, it'll alight. And finally, it will land. Many mosquitoes will start biting right away. Others will walk around for a bit and sample the surface. And the question is, again, are the cues all the same? If I would think back to what I thought was the case with moths, we would say a couple of compounds, those compounds are active all the way through the process. But it's possible that that's not the case. And one of the problems with the papers that are being published right now is they look at a single cue at a time. They look at a visual cue, they look at a heat cue. None of them seem to really do a very good job of looking at an odor cue. And the insect itself is confronted with all of these cues simultaneously, so which are important. So the papers that have been published in Current Biology recently kind of suggest that visual cues and heat cues, visual cues are paramount. So we've looked at this, and one of the students in the lab, Ben Summer, has devised a really nice way of comparing the valence, that is the importance value of cues. Again, it uses a wind tunnel. It pipes carbon dioxide in at the upwind end. Mosquitoes are released at the downwind end. And on the floor, you see a little tray. And actually the stimuli are not colored red or green, they're all black, but one is designed to tell you that you could have a heat cue at 37 Celsius, or you can have an odor cue. And the odor cue is arranged by, I like to call them volunteers in the lab who are willing to wear socks with beads in them for extended periods of time. And that produces a very nice odoriferous bead, which mosquitoes will react to. So what's different about this is that it's asked the question in the sense that it's not whether they will respond to a cue, but given a choice in cues, what is paramount. So Ben's done a lot of work with several thousand mosquitoes. I'm going to show you a couple of results from that, from the most important comparisons. But here's a closer view of the setup here. So you're looking down in the wind tunnel right now, and what you see are the beads. There's a little bit of color there because of the way in which the camera is reproducing. They're all basically black beads. And so what you can have is a section of heat odor, which is fairly small, a couple of inches across. It can't be discerned by visual means because it's buried. Or you can also have a section of skin odor, which again can't be discerned because it's buried. There's a fairly even surface there. And what we were asking questions are all of these comparisons. Do females prefer heat, or do they prefer skin odor? And a lot of other comparisons that we can make. So here's an example of how this plays out. And these are the numbers landing of skin odor versus heat. And actually, the number landing at heat is in some respects perhaps a little higher than we might expect. I'll show you a little bit why, but the number landing on skin odor is in fact very high, and that suggests that heat is not that important compared to skin odor. By the same token, visual cues are not that important relative to either of those cues. Here you have the actual landing times, and you can see again that skin odor provokes an extended period of walking about on the surface and probably probing with the proboscis. You can further look at this by comparing heat versus clean beads, and then you get a very poor reaction. And my suspicion here is that in the previous panel, what you've got a little bit is that they did get some exposure to skin odor when they landed on the heat, so that carries over and keeps them behaving a little bit differently than if they've never had exposure to skin odor. One other thing that's really remarkable about this, I'm going to go back and show you this here, is that, again, visual cues have been surmised to be extremely important, and the mosquitoes, when they go to land on that very small patch of skin odor, are superbly able to find exactly where it is. And you do not need a visual demarcation for them to actually land on it. So that was a great surprise. And it may have some implications from how we set up traps in the future as well. Okay, so here's that, and the time, and a few of them did stay a while on heat, those were there. So the question then arises, do we have any sense for what the chemistry of the odors might be? Remember, I told you at the start there were more than 400 compounds, and there's been a long history of trying to understand what they mean. The most common method that a lot of chemical ecologists use is, was described by Milt Silverstein, one of the founders of chemical ecology, is you run it up the flagpole to see who salutes, meaning you identify a lot of compounds, and then you see if anyone reacts to them. And so this has been the stock and trade of many labs for many years, and you do get some reaction. So that's, I guess, interesting. But my sense is that when you have to wait 7 or 8 minutes for a good capture in one of these bioassays, you really do not know what's going on. So what we thought at that point is that we should maybe use a different kind of bioassay. And remember, I told you with Anopheles gambiae, it was possible to have them landing on pantyhose that one volunteer has worn. And so we went back to our skin odor beads and went through something that was more conventional in terms of old school chemical ecology. meaning we did not try to do empty neurons, informatics, any of the procedures that have been very important, I think, in understanding inputs into the chemical sensing of odors. But we just went back and said, okay, we're going to do fractionation followed by bioassay, followed by fractionation, followed by bioassay. And it turned out to be not that easy to do because of several factors in the way the chemicals behave. But I had the great fortune of taking one of Johnson Miller's PhD students, John Bello, and having him work on this project for almost a year. And you can see on the left, I trust, this particular project here, you can see you get more than half of the mosquitoes landing, spending an extensive period of time on these glass beads, very few landings on the clean beads, and it turns out there is, in fact, a blend of compounds, two of which are new to the mosquito world, which cause this. So one of the things we're trying to understand right now is what other behaviors are evoked by these chemical mixtures. One way that we're going to be doing that is to use, again, the wind tunnel system that we have. Now you're looking down at the wind tunnel, and you can see a section of glass beads elevated from the floor, and the mosquitoes will be given carbon dioxide in advance, and they have a chance to go up and land on it. So we're trying to understand if the compounds mixture that evokes landing, in fact, also evokes attraction. You also might wonder what it evokes besides landing, and so we're also looking at whether or not it does seem to evoke probing behavior. So it seems that there's another question there. Is this being sensed at the tarsal level, the antennal level? At this point, we just don't know. So that's an exciting area, and we hope that this might yield some compounds that are helpful in making traps better. So now I'm going to concentrate on work that has been done rather recently by Brogan Amos, a postdoc who arrived last year, from Australia. And she had the opportunity to work in Scott Ritchie's Queenslander recreation of the outdoors in his lab at James Cook University. And the question that arose here was, in a way, analogous to the one that arises when you say, you go out in the field, Aedes aegypti lands on you very quickly, tries to bite you. But I've heard from many folks who work with traps that, in fact, they don't seem to think that a very large proportion of the animals that are lured to the vicinity of traps actually go in. So we wanted to look at that with the traps that are normally used for surveillance. And we settled on looking at 3 kinds of traps with different configurations. This work actually is already, one of the papers is already out, another one just got accepted today. So these will be available for your perusal. But the idea is to use the Biogents Sentinel trap in various ways to look at the question of, first of all, if you configure it with carbon dioxide, or you configure it with carbon dioxide plus their commercial lure or the commercial lure, how are the efficiencies? And then the other question to ask is, what are the efficiencies? So here, and Brogan can correct me when I'm going off base here. Postdocs here to tell me I made a mistake. So this is the way in which the traps work. You have carbon dioxide that is being forced out, lures being forced out. We use a tank with a regulator so we can have a very precise regulation of carbon dioxide. Clearly, there are visual cues involved. Depending on which trap you look at, there can be black and white areas. And the particular setup in Queensland is this one here. And you can actually see where the video systems are set up. The video is set up on the top, and that's where most of the analysis comes from, the tracks that you can see mosquitoes approaching. They either go over the trap, land on the trap, or perhaps even get trapped. We also have some records from the side that give us a little more detail on how certain mosquitoes behave and why they may or may not get captured. So the way in which this gets done is that, say, 100 male or females are released daily I guess, and this goes on for about a week. And we have an estimate of mortality in the system, so we pretty much know how many animals should be there and how many are available to trap. And the experimental design is pretty straightforward. We have several different kinds of treatments, of course. We have the trap with carbon dioxide plus the lure, the trap with the lure without the carbon dioxide. And of course, you can have it totally unbaited. And you can look at a number of different things. The most important one we're going to look at is the number of encounters versus capture. But we also wanted to look at the flight tracks to see if we could gain some insight as to why some animals may have escaped capture and others got caught. So this is a series of things that we will now sort of go through for, first of all, for females. And what you see here is that the number of encounters is pretty high if you have the carbon dioxide plus the lure. But what you find is that in all cases, the actual capture rate is pretty low. So that's kind of disappointing, I suppose, if you're in a surveillance mode, because the last thing you want is a false negative. And we've actually done quite a bit of work modeling this with moths, and it really is a problem of failing to understand you have a population if you have false negatives. And the problem with almost all of these systems is no one realizes that they have false negatives. So another way you can look at this is to look at the highest proportion captured was, let's see, with the trap alone. Let's see. And there's no difference in the duration of encounter, just a lot of sloppiness in terms of the actual behavior. However, the velocity did differ in one case, but you know, I think it's very hard to make a case that that's of any real behavioral importance. The velocity, it increases over the trap entrance. You might ask the question, could you increase the suction in the trap and get more of them pulled in? Turns out that long ago, Kennedy determined they fly at least 1.5 meters a second, and they are perfectly capable of flying over these traps and not getting sucked in. And so the question really arises, what would cause them to linger long enough and stop over the trap entrance in order to get captured? So we don't seem to have anything that does a good job of that. We're also going to look at the straightness of the track. In other words, possibly if it's more circuitous, zigzaggy, that would favor them getting captured. But again, you don't really see much of a difference here. There was one, but it didn't really, to my way of thinking, it doesn't really tell us very much. And then ultimately, again, this is the trouble reading this here, the proportion of females congregate. This is at near the trap entrance. Yeah, so here you can actually see a nice little track from two sides, but again, it doesn't really tell you very much, I guess, about why any of them are going to be captured or not. Now, this is an example of what you can do if you want to measure the airflow. And what you see here is that air is sort of coming out of this sort of a circular white area surrounding the trap entrance. And so that potentially is attractive, but what it seems to do as much as anything is cause mosquitoes to land on it, rather than to sort of go near the trap entrance. And you can see that there's a higher velocity of the trap entrance, but what we learned is that they just tend to fly over it and rarely get pulled in. Now, the males tell a very parallel story, and why will we be trapping males? Well, there's a lot of interest in this for a lot of the manipulations being genetic modification of the males, also with Wolbachia, et cetera, to find out where are they. And so that's unusual compared to most other mosquitoes. And these mosquitoes are also unusual in that males congregate near where females are apt to be feeding on a host. So it's not surprising that you would in the field capture them. And of course, we release them here in the same way we did with females. And you will see a parallel story here that tells us relatively little, except that the actual number that are captured are really minuscule. So we're really talking maybe in the best case of what, maybe 10 or 12%, and but generally speaking, it's just not a very satisfying story if you're interested in capturing a significant portion of the males. So we can go through these. You can see some of the behaviors that sometimes occur. They don't seem to go near the trap entrance in the same way. They seem to hover over the area producing the lure. So that's somewhat different, but eventually some of them will sort of venture in a place that they shouldn't and they will get captured. But in fact, the behavior is a little bit different here. And again, without spending a lot of time on it, there are some differences that you can see, and in particular, again, that draw your attention to these behaviors near the trap. By the way, that white, this is a little bit farther view of it, that white area is where we actually started tracking the behavior of males or females. So that's below the trap, and it's a piece of white plastic. And again, here's a side view, and you see a male that is very capable of flying very close to where the suction is, and suction is insufficient to bring him in. If we were to imagine that suction would be greatly increased, I'm not sure how the trap would perform. You would have to probably change the white surface that is allowing the air and the lure to come out that at the periphery. Not really sure how that would work. So the conclusions that one can draw from this, and I should point out that actually there's one more study that's been written up and submitted, and that's using the CDC style trap, which probably is what a lot of people are using in capturing Aedes aegypti with. And we find, I'm afraid, exactly the same thing. Very, very low capture efficiency, and no prospect that would be highly valuable per se for surveillance. So it's true that these have low efficiency. Mosquitoes are attracted to the trap. Many rest on the trap. It's kind of disturbing that you see them sitting on the trap rather than getting captured. We speculate it's certainly conceivable you might put an insecticide on there to trap them, but that's probably not something that's going to happen either. So sort of going forward, there are three things that we will probably continue to do. The first of which is behavioral, and we will continue to look at those compounds that are involved in landing and probing to see whether they're attractive or whether we have to add other compounds to get a better lure. So that's consuming part of our lab at this point. A second area that certainly we'll be spending a lot of time on, we're going to be spending a lot of time on this year, is to take these data, which are, we call semi-field data, and go out in the field and verify that more or less the same thing is going on in terms of efficiency in the field. I would be very surprised if it was not identical, because we can watch these things come up to the trap, obviously lured to the trap's vicinity, but they don't get captured. I wouldn't expect that would differ in the field, but that needs to be done. And then the third area is that given that we understand a little bit about why animals are not captured, we think we can perhaps find ways to modify the configuration of traps such that mosquitoes that venture near the suction, in fact, may continue on and actually get captured. Whether that's a design of the trap and or changing the chemistry of what we put out in the trap, or both, remains to be determined. So that's pretty much what I have to tell you. I guess there's time for questions. Oh, I have some acknowledgments. I always forget the acknowledgments. Let's do that. There are a lot of folks. Scott Ritchie and his colleagues at James Cook University. It was funded, of course, by the TACVET group, which we appreciate. Here at my group, there are several folks who have helped out. We have Emerson Lacey, Megan Moore, and I've had a lot of past folks who've been important in all of this. Ben Decker, who did his degree with mostly Aedes aegypti work. Martin Geyer came to my lab in 1997, spent a year working on wind tunnels and trying to understand the behavior of aegypti. Of course, he now runs the company that sells the Biogents. Marita Brax, in collaboration with Walter Leal, looked at overposition stimuli for Culex. And Miriam Cooperband also ran an elegant series of studies on trapping of Culex, Quince fasciatus, and Tarsalis to show pretty much the same thing. The traps don't work very well using a large field wind tunnel. And Ben Webster was an excellent postdoc who was willing to wear pantyhose and all kinds of things for me that I would not be willing to do myself. And Jocelyn Miller had given some advice on the chemistry, and I've worked with Anand Ray on several of these projects as well, and Walter Leal. Thanks for reminding me of that.